Chapter 1: Cool down and quieting of muscle. The workout has ended. The world inside your muscle is dim now. Warm, quiet. We are floating inside a single fiber of your quadriceps. The contraction has stopped. The demand has lifted. Motor units are de-recruiting. The nerves that fired during each rep are going silent. One by one, the neural drive to the muscle is fading. Rapidly, within moments, the command to contract is gone. The muscle remembers the work, but the urgency has passed. We drift along the sarcolemma now. This is the outer membrane of the muscle cell. During exercise, ions moved across this barrier. Sodium rushed in, potassium leaked out. The membrane became excitable, electric. Now the restoration begins. The sodium-potassium pump is working. Quietly, it gathers sodium from inside the cell. It moves sodium back outside. It brings potassium back in. 3 sodium out, 2 potassium in. Again and again, this pump is a night custodian. It walks the electrical hallway. It returns each ion to its proper place. Order is being restored. The ion gradients are stabilizing. The membrane is calming. Excitability is normalizing. This happens in darkness, in silence, only chemistry, steady and slow. We descend into the T-tubules now. These are narrow tunnels that reach deep into the fiber. They carry the signal to contract. Now they carry stillness inward. The sarcoplasmic reticulum is near. This is where calcium is stored. During each contraction, calcium flooded out into the fiber. It allowed the muscle to shorten, to grip, to pull. Now the calcium is being gathered. Circa is the name of the pump. It moves along the reticulum like a librarian. It collects calcium ions. One at a time, it shelves them back into storage. The reading hall empties. The space becomes still. As calcium is resequestered, the muscle releases, the fibers relax, the grip loosens. Excitability normalizes. The tension dissolves into the dim warmth. We move deeper now, into the mitochondria. These are the power centers of the cell. Small, dark. They float in the cytoplasm like lanterns in a quiet room. During exercise, they produced ATP constantly. They burned through fuel. They kept the muscle moving, but they also created phosphocreatine. Phosphocreatine is a fast reserve, a rechargeable battery. It stores energy in a single bond. During the workout, that bond broke. The energy was released. ATP was remade instantly. Now, the refilling begins. Phosphocreatine resynthesis starts within seconds. The mitochondria take oxygen, they take creatine, they take ADP, they rebuild the bond. The battery recharges, the lantern refills its light, slowly, evenly, without urgency. This happens in a pattern. The recovery follows what is called mono-exponential kinetics. It is smooth, predictable, fast at first, then slower. The time constant is about 30 to 60 seconds. In that first minute, half of the phosphocreatine returns. Most of it returns within a few minutes. The speed of this depends on mitochondrial oxidative capacity. If the mitochondria are trained, the refilling is quick. If they are not, it takes longer. Your muscle knows its rhythm. As phosphocreatine is resynthesized, something else happens. The pH begins to recover. During exercise, hydrogen ions accumulated. The environment became acidic, sharp. Now, the acid is clearing. The hydrogen ions are buffered, moved away. The pH drifts back toward neutral. This recovery is coupled to phosphocreatine resynthesis. The two processes move together. As the battery recharges, the acid fades. They hold hands in the dark. We float outward now, toward the capillaries. These are the smallest blood vessels. They wrap around the muscle fiber like fine threads. During exercise, they delivered oxygen. They removed waste. The work has stopped. But blood flow has not. The vessels remain open, dilated. Blood continues to move through them. This is called reactive hyperemia. It is a lingering flow. A river that keeps moving after the work is done. The blood rinses the tissue. It brings oxygen. It carries away lactate. It carries away potassium. It carries away hydrogen ions. The capillaries do not close suddenly. They ease back, gradually, patiently. Near-infrared spectroscopy can watch this. NIRS measures oxygen in the muscle. During exercise, oxygen levels fell. The tissue used oxygen faster than it could arrive. Now, microvascular oxygen saturation is normalizing. The tissue is reoxygenating rapidly. The balance is shifting. Oxygen delivery is rising. Oxygen utilization is falling. The two meet in equilibrium. The muscle breathes again. We sense the temperature now. The fiber is warm, warmer than before the workout began. Exercise created heat. Chemistry created heat. Contraction created heat. But the heat is leaving now. Intramuscular temperature is beginning to decline. Slowly, over the first hour, it will drift back toward baseline. The muscle cools like embers fading in a hearth. As the temperature drops, the chemistry slows. Enzyme kinetics are temperature dependent. When warm, they move quickly. When cool, they slow down. Now the enzymes are easing into a calmer metabolic pace. The reactions become gentler, less urgent, more measured. The warmth leaves the room. The chemistry follows. There is something else happening as the muscle cools. The viscoelastic elements are changing. During exercise, the tissue was compliant, soft, pliable. Now there is a subtle shift, a gentle return toward resting firmness. Not stiffness, just a settling. The warm state is fading. The resting state is returning. The tissue remembers its shape. We move into the interstitial space now. This is the fluid between muscle fibers. It holds water, ions, metabolites, small molecules. During exercise, this space became crowded. Lactate pooled here. Hydrogen ions accumulated. Potassium leaked out from the fibers. Now the clearing begins. Blood flow is ongoing. Lymphatic vessels are open. The metabolites are being drained away. Lactate is carried off. Hydrogen ions are buffered and removed. Potassium is reabsorbed. The interstitial space is lightening, becoming transparent again, clean. The fluid flows slowly, steadily, without rush. Above all of this, the autonomic nervous system is shifting. During exercise, the sympathetic branch was dominant. It drove the heart faster. It opened blood vessels wide. It pushed the body forward. Now, parasympathetic influence is rising. This is the branch of rest, of stillness, of repair. It does not arrive suddenly. It arrives like twilight. Gradually, gently, this shift shapes the microcirculation. Blood flow does not stop abruptly. It tapers. The vessels constrict slowly. Perfusion reduces in a controlled way. The muscle is not abandoned. It is guided into quiet. We pause here. Floating in the dim warmth, the fiber is cooling. The ions are balanced. The calcium is stored. The phosphocreatine is refilling. The blood is clearing waste. The autonomic tone is shifting toward rest. The muscle is no longer working, but it is not yet fully at rest. It is in between, in the dimming, in the cooling, in the clearing. This quiet is necessary. It creates space. It allows repair signals to begin. The next phase will emerge from this stillness, but that comes later. For now, there is only this. The slowing, the softening, the return toward baseline. The muscle fiber drifts in the dark. The chemistry eases. The warmth fades. The tension dissolves. Everything here is becoming still. Microdamage and early repair. Signal. The hours have passed. We are still inside the muscle. Deeper now, closer to the smallest structures. The cooling has finished. The metabolites have cleared. The fiber is quiet, but something has changed. We drift toward a single sarcomere. This is the repeating unit of contraction. During the workout, this sarcomere shortened and lengthened thousands of times. It pulled, it resisted, it absorbed force. That force left marks. We see the Z discs first. These are the boundaries of the sarcomere, dark lines that anchor the thin filaments. Normally, they are crisp, organized, parallel. Now some of them are streaming, blurred. The lines are not straight. They waver. This happens after unaccustomed or high-tension eccentric contractions. When the muscle lengthens under load, the Z discs absorb strain. Sometimes they distort. Biopsies can capture this. Electron microscopy can magnify it. The disruption is real, measurable, visible, but it is not catastrophic. It is a signal. We move along the sarcomere now. past the Z disc, into the center. Here, titin stretches between the thick filament and the Z disc. Titin is a giant spring protein. It provides passive tension. It keeps the sarcomere organized. After eccentric work, titin can be disrupted. The disruption occurs at the mid-sarcomere. The spring loses some of its structure. The sarcomere becomes non-uniform. Some regions stretch more than others. The symmetry is broken. This is temporary. The fiber will rebuild. But for now, the architecture is disturbed. We notice the Desmond network next. Desmond is a protein that links sarcomeres to each other. It connects Z discs across the width of the fiber. It provides lateral support. It maintains alignment. After intense contractions, the Desmond network can become disorganized. The connections loosen. The alignment shifts. This is especially pronounced in fast-twitch fibers. They contract with more force. They accumulate more strain. The Desmond will reassemble, but the disturbance is part of the early repair signal. The cell senses this disorganization. It responds. We pull back slightly now, away from the sarcomere, toward the T-tubules and sarcoplasmic reticulum. These structures form what is called a triad. The T-tubule in the center, the sarcoplasmic reticulum on either side. This is where excitation-contraction coupling happens. The electrical signal arrives at the T-tubule. Calcium is released from the reticulum. The muscle contracts. After heavy eccentric work, this coupling can be perturbed. The triad can become uncoupled. The signal does not translate as cleanly into contraction. This is called low-frequency fatigue. The muscle loses force production, especially at low stimulation frequencies. The machinery is still intact, but the communication is disrupted. The signal weakens before it reaches the contractile proteins. This takes time to repair, days sometimes. But the repair will come. We drift toward the sarcolemma now, the outer membrane. During the workout, this membrane stretched and deformed with each contraction. In some places, the strain was too much. Sarcolemmal permeability has increased. Small molecules can pass through the membrane more easily now. In animal studies, researchers inject Evans blue dye into the bloodstream. The dye binds to albumin. Normally, it stays outside the muscle fiber, but when the membrane is disrupted, the dye enters. The fiber takes on a blue tint. This is how scientists measure membrane damage. It is a visible proof. The damage is sublethal. The fiber has not died, but it has been compromised. This triggers repair signaling. The cell detects the breach. It begins to respond. We move outward now, into the extracellular matrix. This is the scaffolding around the muscle fiber. It is made of collagen, proteoglycans, structural proteins. It holds the fibers in place. It transmits force between them. After exercise, this matrix begins to remodel. Enzymes are released. Matrix metalloproteinase 9 is one of them. MMP9 breaks down damaged collagen. It clears space for new matrix to be laid down. Tissue inhibitor of metalloproteinase 1 also rises. TIMP1 controls MMP9. It prevents excessive breakdown. The two work together, breaking down, building up, a controlled cycle. Later, MMP2 and IMP2 will appear. They refine the remodeling, they adjust the structure. The matrix is not static, it adapts. The connections between the fiber and the matrix are changing too. Integrin alpha 7, beta 1 is a receptor on the sarcolemma. It links the inside of the cell to the extracellular matrix. It is part of a structure called the costamere. After training integrin alpha 7, beta 1 expression can rise. The costameres are strengthened. The mechanotransduction pathways are enhanced. The fiber becomes better at sensing and transmitting force. Focal adhesion kinase is activated as well. FAK is a signaling molecule. It sits at the junction between the cell and the matrix. When force is applied, FAK phosphorylates. It sends signals inward. It tells the cell that mechanical load has occurred. This is how the muscle knows to adapt. The force is sensed. The signal is transmitted. The response begins. You notice movement now. Small cells are arriving. They are immune cells, neutrophils. They are the first responders. Within hours of the workout, they accumulate in and around the damaged muscle. They move through the capillaries. They squeeze between endothelial cells. They migrate toward the site of disruption. They are drawn by chemical signals released from the injured tissue. Neutrophils clear debris. They remove damaged proteins. They prepare the space for repair, but they do not stay long. Their role is brief, necessary but temporary. Behind the neutrophils, macrophages are arriving. They follow a few hours later. Macrophages are larger, more versatile. They have multiple roles. Some macrophages are pro-inflammatory. They continue the clearing work. They break down damaged structures. They release signals that promote inflammation. Other macrophages are anti-inflammatory. They arrive later. They dampen the inflammatory response. They release signals that promote tissue repair. They encourage regeneration. The balance between these macrophage subsets shapes the transition from inflammation to repair. It is a choreography, timed, controlled. Regulatory T cells are present as well. They are part of the adaptive immune system. They do not attack the muscle. They modulate the environment. They help maintain a controlled inflammatory response. They prevent excessive damage from the immune system itself. The immune presence is not an invasion. It is a collaboration. The cells work together to restore the tissue. We notice something else now, a sensation, soreness. It has been building since the workout ended. By now, it is present, dull, deep, tender to touch. This is delayed onset muscle soreness, DOMS. It peaks around 24 to 72 hours after exercise. It is not caused by toxins. This is important. There are no toxins accumulating in the muscle. The soreness is driven by nociceptor sensitization. Nociceptors are sensory nerve endings. They detect tissue damage. They respond to chemical signals released during inflammation. Bradykinin is one of those signals. It is a peptide. It is released during tissue disruption. It binds to receptors on nociceptors. It lowers their threshold. They become more sensitive, more reactive. Prostaglandins contribute as well. They are produced by cyclooxygenase enzymes. COX2 is upregulated after exercise. It produces prostaglandins that sensitize nociceptors. They amplify the pain signal. Nerve growth factor and glial cell, line-derived neurotrophic factor, also participate. NGF and GDNF are released during muscle damage. They enhance nociceptor sensitivity. They prolong the soreness. Edema plays a role too. Fluid accumulates in the interstitial space. The tissue swells. The swelling creates mechanical pressure. It activates high-threshold mechanoreceptors. Thus, receptors normally respond only to strong pressure, but when the tissue is swollen, they become active at lower thresholds. The soreness is a signal. It is the nervous system's way of communicating that repair is underway. It encourages rest. It discourages excessive movement. It is protective, but it is not damage itself. The soreness is the sensation of repair signaling, nothing more. We pause here, floating in the tissue. The sarcomeres are disrupted but not destroyed. The membrane is permeable but repairable. The matrix is remodeling. The immune cells are working. The nociceptors are sensitized. This is not a crisis. This is the beginning of adaptation. The muscle has been challenged. It has been marked by that challenge. Now it is responding. The signals are spreading. Inside the fiber, genetic programs are activating. Transcription factors are moving into the nucleus. Repair pathways are being initiated, but those pathways require energy. They require fuel. They require time. That fuel will be restored during the night. Glycogen will be resynthesized. Mitochondria will adapt. The energy systems will rebuild, but that comes next. For now, there is only this. The quiet assessment, the immune choreography, the signaling cascade, the soreness that reminds the body to rest. The muscle is damaged, but the damage is purposeful. It is the beginning of something stronger. Everything here is preparing to rebuild. Chapter 3, Evening Night, Energy and Mitochondrial Recovery. The sun has set. The body is still. Resting, the muscle fiber is quiet now. The soreness lingers, but the immune cells are working. The repair signals have been sent. Now Now the night shift begins. We drift inside the cell. The cytoplasm is dark, warm. There is no urgency here, only maintenance. Only the steady work that happens when the body is at rest. Energy is being managed. ATP is the currency of the cell. It powers everything: protein synthesis, ion pumping, membrane repair, movement of molecules. Even in stillness, the cell requires ATP. The demand never stops, it only slows. During the night, mitochondria continue their work. They produce ATP steadily, quietly. They take oxygen, they take fuel, they convert it to energy. The production is not frantic, it is measured, sufficient. This ATP supports everything happening in the recovery process. The ion pumps that restored balance after exercise are still running. The sodium-potassium pump, the calcium pump, they require ATP to function. They never rest completely. Protein synthesis is accelerating now. Ribosomes are assembling amino acids into new proteins. This requires energy. ATP is consumed with each peptide bond formed. The cell is rebuilding what was damaged. It is reinforcing what was stressed. Phosphocreatine is being maintained as well. The stores that refilled in the first minutes after exercise are now stable, but they require ATP to stay full. The mitochondria provide that ATP. The balance is preserved. High energy phosphate ratios remain relatively stable during these sustained recovery phases. The cell is not depleting its reserves. It is maintaining them. The energy system is in equilibrium. This is the quiet work of ATP homeostasis. It happens in darkness, without fanfare. It is simply the cell taking care of itself. We move toward the glycogen granules now. These are clusters of glucose molecules. They are stored inside the muscle fiber. During exercise, they were broken down. They provided fuel for contraction. They were depleted. Now, they are being rebuilt. Glycogen resynthesis follows a pattern, a biphasic pattern. There are two phases. The first phase is rapid. It begins immediately after exercise ends. It continues for a few hours. This early phase does not depend heavily on insulin. The muscle is primed. Contractions have sensitized the fiber to glucose uptake. Glucose moves into the cell without much hormonal assistance. It is converted to glycogen quickly. Glycogen synthase is the enzyme responsible. It adds glucose molecules to the growing glycogen chain. After exercise, glycogen synthase activity is high. The enzyme works efficiently. The stores refill rapidly. But this rapid phase does not last forever. The second phase begins as evening turns to night. This phase is slower. It continues through the night. It can last many hours. It depends more on insulin. As glycogen stores begin to normalize, the urgency fades. The rapid refilling slows. Glycogen synthase activity gradually declines. The enzyme is still working, but it works more slowly now. Insulin begins to play a larger role. During the day, after meals, insulin rises. It signals the muscle to take up glucose. It enhances glycogen synthesis. The hormone guides the slower, sustained phase of refilling. By morning, if nutrition has been adequate, the glycogen stores are approaching normal levels. The granules are full again. The fuel reserve is restored. AMPK is involved in this process. AMPK is a cellular energy sensor. It activates when energy is low. During and after exercise, AMPK is highly active. AMPK enhances glucose uptake. It increases the number of GLUT4 transporters on the cell membrane. GLUT4 is a glucose transporter. It allows glucose to enter the muscle fiber. When AMPK is active, more GLUT4 appears. More glucose enters. Glycogen can be resynthesized. But AMPK has another role. When glycogen stores are ample, AMPK can inhibit glycogen synthase. It prevents overfilling. It maintains balance. As the night progresses, AMPK activity falls. The energy stress has passed. The cell is no longer in deficit. AMPK steps back. As AMPK activity declines, insulin's influence predominates. The handoff is complete. AMPK managed the early recovery. Insulin manages the late recovery. The two hormones work in sequence, not in competition. This is how the cell restores its fuel across time, rapidly at first, then slowly, guided by signals that shift as the hours pass. We descend into the mitochondria now. These organelles have been working all night, producing ATP, supporting recovery, but they are also being evaluated. Exercise creates stress on mitochondria. They produce reactive oxygen species. They accumulate damage. Some mitochondria become dysfunctional. Their membranes depolarize. Their efficiency declines. The cell has a system for this: mitophagy. Mitophagy is the selective removal of damaged mitochondria. It is a form of autophagy. The cell engulfs the dysfunctional organelle. It breaks it down. It recycles the components. After exercise, mitophagy can be triggered. AMPK plays a role here as well. AMPK activates ULK1. ULK1 is a kinase that initiates autophagy. The pathway begins. Parkin is another protein involved. When a mitochondrion is damaged, parkin translocates to its outer membrane. Parkin tags the organelle for degradation. The autophagosome forms around it. The mitochondrion is consumed. This happens slowly, over hours, not immediately after exercise. The process continues through the evening and into the night. During sleep, mitophagy progresses. The cell clears what is broken. It makes space for what is functional. Mitochondrial quality control is ongoing. It is part of the maintenance work that happens when the body is at rest. The mitochondrial network itself is changing. Mitochondria are not static. They fuse and divide. They form networks. They fragment into smaller units. The shape of the network reflects the metabolic state of the cell. During exercise, mitochondria can fragment. They become smaller, more numerous. This supports rapid ATP production. Later in recovery, the network can shift toward a more fused morphology. The mitochondria elongate. They connect. Fusion increases efficiency. It allows the sharing of components. It stabilizes function. This remodeling happens gradually. It is most pronounced during rest and sleep. The cell is not being disturbed by contraction. The mitochondria can reorganize without interruption. The network becomes more efficient, more resilient, better prepared for the next bout of exercise. Reactive oxygen species are present throughout this process. ROS are byproducts of mitochondrial metabolism. They are often thought of as harmful, as damage-causing molecules, but they are also signals. After exercise, ROS levels rise. They are produced during intense ATP production. They leak from the electron transport chain. They interact with cellular components. The cell detects this. It responds. It upregulates antioxidant enzymes: superoxide dismutase, catalase, glutathione peroxidase. These enzymes neutralize ROS. They protect the cell from oxidative damage. But this upregulation does not happen instantly. It takes time. The genes must be transcribed. The mRNA must be translated. The proteins must be assembled. Much of this work occurs during rest. During the night, the transcription and translation consolidation happens when the cell is not being stressed by contraction. Over repeated recovery cycles, the antioxidant capacity increases. The cell becomes more resistant to oxidative stress. This is adaptation. ROS act as the signal that triggers it. The initial stress provokes the response. The response makes the cell stronger. This is how training works at the molecular level. We pause here. Floating in the cytoplasm, the mitochondria are producing ATP steadily. The glycogen granules are refilling. AMPK activity is fading. Insulin influence is rising. Damaged mitochondria are being cleared. The network is remodeling. Antioxidant enzymes are being synthesized. All of this happens in darkness, in stillness, without conscious effort. The muscle fiber is repairing itself. It is restoring its energy reserves. It is upgrading its infrastructure. It is preparing for the next demand. But there is more happening beyond the muscle. The nervous system is also shifting. The autonomic tone is settling. The body is entering deeper rest. That transition is quiet, gradual. It shapes how the entire organism recovers. But that comes next. For now, there is only this: the night shift inside the cell, the steady production of ATP, the slow refilling of glycogen, the careful maintenance of mitochondria, the adaptive response to oxidative signals. The muscle is not sleeping, but it is resting, rebuilding, becoming stronger in the dark. Everything here is working toward balance. Chapter 4. Nervous system, downshifting, and muscle recovery. The night deepens. Inside the muscle, the work continues. ATP is being produced. Glycogen is refilling. Mitochondria are remodeling. But the muscle does not control itself alone. It is guided by the nervous system. We pull back now, away from the fiber, away from the cell. We drift upward, through the body, toward the control room. The nervous system is dimming its lights. After exercise, the heart was beating quickly. Blood was pumping hard. The sympathetic nervous system was active. It drove the body forward. It sustained the effort. Now, the effort has ended. The heart begins to slow. This happens in 2 phases. The first phase is rapid. Within the first minute after exercise stops, heart rate falls quickly. This is driven by parasympathetic reactivation. The vagus nerve wakes up. It releases acetylcholine at the heart. The signal is clear. Slow down. The heart obeys. The rate drops. The pounding eases. The second phase is slower. Over the next several minutes, sympathetic withdrawal occurs. The adrenaline fades. The norepinephrine clears. The drive to maintain a high heart rate diminishes. The heart continues to slow. Gradually, steadily. By the time evening arrives, the heart rate has settled near its resting baseline. The urgency is gone. The rhythm is calm. This recovery is not just about the heart. It is about the entire autonomic nervous system. The balance between sympathetic and parasympathetic tone is shifting. The body is moving from alertness toward rest. Heart rate variability reflects this balance. HRV measures the variation in time between heartbeats. When parasympathetic tone is high, HRV is high. The heart is flexible, responsive, adaptive. When sympathetic tone is high, HRV is low. The heart is rigid, driven, fixed. During deep NREM sleep, vagal-related HRV indices are especially high. The parasympathetic system is dominant. The vagus nerve exerts strong influence. The heart rate slows. The variability increases. This is associated with better cardiovascular status, better recovery. The body is in a restorative state. The nervous system is allowing the heart to rest. Athletes with higher HRV during sleep tend to recover more completely. The autonomic system is functioning well. The balance is healthy. We notice something else now. The body is cooling. Core temperature is dropping. This happens before sleep begins. It is part of the sleep onset process. The body needs to cool in order to fall asleep deeply. This cooling is driven by peripheral vasodilation. The blood vessels in the hands and feet open. Blood flows toward the skin. Heat radiates away from the core. The internal temperature falls. The magnitude of this core temperature drop matters. Larger drops correlate with more consolidated NREM sleep. The body cools. The sleep deepens. The two are linked. The temperature drop also correlates with increased nocturnal HRV. As the core cools, parasympathetic tone rises. The vagus nerve becomes more active. The heart rate variability increases. The body is coordinating multiple systems. Thermoregulation, autonomic tone, sleep architecture. They work together. They support each other. The cooling continues through the night. The lowest core temperature occurs in the early morning hours. By then, the body has been in deep rest for hours. The restorative work is well underway. We descend into the spinal cord now. This is where reflexes are processed. Reflexes are automatic responses. They do not require conscious thought. A tap on the knee. The leg kicks. This is a reflex. The H reflex is one such response. It is a stretch reflex. It involves the connection between sensory neurons and motor neurons in the spinal cord. Researchers use it to measure spinal excitability. During wakefulness, the H reflex is strong. The spinal cord is responsive. The reflex arc is active. During NREM sleep, something changes. The H reflex is depressed. The gain is reduced. The spinal cord is less excitable. The reflex is quieter. This is not dysfunction. This is rest. The nervous system is downshifting. The reflexes do not need to be as sharp during sleep. The body is not moving. The environment is safe. The vigilance can decrease. Nociceptive reflex thresholds also increase during NREM sleep. Nociceptive reflexes are pain reflexes. They protect the body from harm. During the day, they are sensitive. A sharp stimulus causes a withdrawal. At night, the threshold rises. A stronger stimulus is required to trigger the reflex. The nervous system is less reactive to pain signals. The body is less easily disturbed. This has implications for muscle recovery. Reduced reflex gain and higher pain thresholds can lower baseline muscle tone. The muscles are not held in constant readiness. They are not bracing. They are not guarding. When muscles are sore, there is often a protective guarding response. The nervous system tenses the muscle to prevent further injury. This tension can persist even at rest. During deep sleep, this guarding softens. The reflex gain is reduced. The pain threshold is higher. The muscle can relax more fully. Blood flow is not restricted by tonic contraction. Recovery is not hindered by unnecessary tension. The nervous system is allowing the muscle to let go. There is another factor at play. Chronic exercise training lowers resting muscle, sympathetic nerve activity. This is an adaptation. Over weeks and months, the sympathetic drive to skeletal muscle decreases. Lower muscle sympathetic nerve activity reduces tonic vasoconstrictor drive. The blood vessels in the muscle are not held in a state of mild constriction. They are more open, more compliant. This may improve nocturnal perfusion of recovering tissue. During the night, blood flow to the muscle is not restricted by sympathetic tone. Oxygen delivery is better. Nutrient delivery is better. Waste removal is better. The trained body supports its own recovery more effectively. The nervous system has adapted. It no longer needs to maintain high sympathetic tone at rest. We pause to consider what happens when sleep is disrupted. Sleep deprivation shifts the balance. When sleep is insufficient, vagal tone decreases. Parasympathetic influence weakens. The body does not rest as deeply. At the same time, sympathetic activity elevates. The body remains in a state of mild alertness. Heart rate variability decreases. The heart is less flexible, less adaptive. This autonomic shift interacts with other systems. Inflammatory markers can worsen. Cytokines rise. The immune response becomes less controlled. Metabolic markers are affected as well. Glucose regulation becomes less efficient. Insulin sensitivity decreases. The body struggles to restore energy balance. Sleep fragmentation has similar effects. Even if total sleep time is adequate, if the sleep is broken, the recovery is compromised. The body cycles through stages less smoothly. Deep NREM is shortened. REM is disrupted. The autonomic system does not settle fully. The parasympathetic dominance of deep sleep is not achieved. The restorative processes are incomplete. This is not a warning. It is simply an observation. The nervous system requires uninterrupted rest to support recovery. When that rest is absent, the support weakens. We drift back now, toward the muscle, toward the fiber. The nervous system has done its work. The heart rate has slowed. The vagal tone has risen. The core temperature has dropped. The reflexes have quieted. The sympathetic drive has withdrawn. All of this creates an environment where recovery can proceed without interruption. The muscle is not isolated. It is embedded in a body. That body is regulated by the nervous system. When the nervous system downshifts, the muscle benefits. Blood flow improves. Tension releases. Pain signals are muted. The next hours will be critical. During slow-wave sleep, growth hormone will pulse. Structural remodeling will accelerate. The deepest repair work will unfold. But that comes next. For now, there is only this. The dimming of the control room, the slowing of the heart, the cooling of the core, the quieting of the reflexes, the release of sympathetic grip. The nervous system is allowing the body to rest fully, deeply, without reservation. Everything here is settling into stillness. Chapter 5. Deep sleep, SWS, GH, and structural remodeling. The body is sinking. Consciousness is fading. The eyes are closed. The breathing is slow. The heart rate is low. The muscles are still. We are descending into deep sleep. This is slow wave sleep, SWS, the deepest layer of REM sleep. The brain waves are long and slow. Delta waves, they roll through the cortex like tides. Heavy, rhythmic, uninterrupted. This is where the heaviest repair work begins. The body has been preparing for this. The core temperature has dropped. The sympathetic tone has withdrawn. The parasympathetic system is dominant. The reflexes are quiet. Now, the hormonal environment shifts. Shortly after sleep onset, a pulse of growth hormone is released. This is the largest daily GH pulse. It occurs during the first cycle of deep sleep. The timing is precise. The magnitude is substantial. Growth hormone is released from the pituitary gland. It enters the bloodstream. It travels through the body. It reaches the tissues, the muscles, the bones, the connective tissue. This pulse is strongly associated with slow wave sleep. When SWS is deep and consolidated, the GH pulse is robust. When SWS is fragmented or shortened, the pulse is attenuated. The body requires this signal. Growth hormone supports tissue growth and repair. It is not the only signal, but it is important. GH does not act alone. It influences the production of insulin-like growth, factor 1, IGF-1. This factor is produced locally in tissues. In the muscle, IGF-1 expression rises in response to growth hormone. IGF-1 promotes protein synthesis. It activates signaling pathways inside the muscle fiber. It encourages the ribosomes to work. It supports the rebuilding process. The presence of growth hormone during early night SWS creates a window where the muscle is primed for repair. At the same time, cortisol is being suppressed. Cortisol is a catabolic hormone. It breaks down tissue. It mobilizes energy. During the day, cortisol rises and falls in a rhythm. It peaks in the morning. It declines through the day. During early slow-wave sleep, cortisol secretion is suppressed. The levels are low. The catabolic pressure is reduced. The muscle is not being broken down. It is being preserved. This combination matters. Growth hormone rising, cortisol falling, IGF-1 being expressed. The hormonal environment shifts toward tissue building, away from tissue breakdown. The muscle protein balance is affected. Protein balance is the difference between protein synthesis and protein breakdown. When synthesis exceeds breakdown, the balance is positive. The muscle grows. The structures are reinforced. During deep sleep, this balance shifts towards synthesis. The anabolic signals are strong. The catabolic signals are weak. The muscle takes advantage of this window. It builds. This happens quietly, slowly, over hours. The ribosomes assemble amino acids. Peptide bonds form. Proteins are created. The damaged fibers are repaired. The structures are strengthened. The body knows how to do this. It has done this every night for as long as the person has been alive. The process is automatic, unconscious. But it is not optional. Evidence for this comes from sleep deprivation studies. When sleep is withheld, the repair is compromised. One night of total sleep deprivation reduces postprandial myofibrillar protein synthesis by approximately 18% the next day. Postprandial means after a meal. Myofibrillar means the contractile proteins of the muscle. Actin, myosin, the proteins that generate force. After sleep loss, the muscle does not respond as well to nutrition. Amino acids are consumed, insulin rises, but the synthesis rate is blunted. The anabolic response is weaker. This is not a permanent impairment. Recovery sleep can restore function, but it demonstrates the necessity of sleep. Normal sleep is necessary to maintain an anabolic response after exercise. Without sleep, the rebuilding slows. The signals are present. The nutrients are available. But the machinery does not operate at full capacity. Nutrition itself plays a role during the night. Pre-sleep protein intake can enhance overnight myofibrillar protein synthesis. When protein is consumed before bed, amino acids are available during the hours of sleep. The muscle uses those amino acids. The synthesis rate increases. The rebuilding is supported. This has been measured. The effect is modest but real. The type of protein matters less than its availability. Casein, whey, whole foods. What matters is that amino acids are present during the overnight period. The muscle can access them. The ribosomes can use them. Not all proteins in the muscle are synthesized at the same rate. Myofibrillar proteins turn over relatively quickly. They can be rebuilt within days or weeks. The muscle is responsive to training and nutrition. Intramuscular collagen synthesis is different. Collagen is part of the connective tissue. It surrounds the muscle fibers. It forms the tendons. It provides structural support. Collagen synthesis appears slower. It is less acutely nutrition sensitive than myofibrillar synthesis. The remodeling timeline is more prolonged. Weeks or months rather than days. This means connective tissue repair lags behind muscle fiber repair. After intense training, the fibers may recover quickly, but the tendons and fascia may still be remodeling. They require more time. This is why patience is important. The muscle may feel ready, but the connective tissue may not be fully adapted. Continued training without adequate rest can stress the slower healing structures. The body coordinates these processes, but it cannot accelerate them beyond their natural pace. There is another layer to this, the unfolded protein response, the UPR. This is a cellular mechanism that handles protein folding stress. Exercise induces stress. Proteins are damaged. New proteins are synthesized. Some of them fold incorrectly. The UPR detects misfolded proteins. It activates chaperones. It slows protein synthesis temporarily. It degrades proteins that cannot be corrected. It protects the cell from accumulating damaged material. The UPR is part of the adaptive response to training. It helps the cell manage the increased demand for protein production. It ensures quality control. Sleep restriction can attenuate the UPR. When sleep is insufficient, the activation of UPR pathways is blunted. The cell is less able to handle protein folding stress. The quality control is compromised. Aging has similar effects. In older individuals, the UPR activation is often attenuated. The coupling between growth hormone and slow-wave sleep can also weaken with age. The GH pulse becomes smaller. The SWS becomes less deep. The combination may reduce effective structural remodeling capacity. The signals are weaker. The quality control is less robust. The rebuilding is slower, less complete. This is not failure, it is a shift. The body still repairs, but the efficiency declines. More time may be required, more care may be needed. We pause here, floating in the darkness. The brain is producing delta waves. Growth hormone has pulsed. Cortisol is suppressed. IGF-1 is being expressed. Protein synthesis is elevated. Protein breakdown is reduced. The balance is positive. The ribosomes are working. Amino acids are being assembled. Myofibrillar proteins are being created. Collagen is being synthesized more slowly. The UPR is managing protein folding stress. The muscle is rebuilding. All of this happens during deep sleep, the heaviest, quietest, most restorative layer of the night. The muscle does not know it is being repaired. There is no conscious awareness, no sensation, only chemistry, only biology, only the automatic processes that sustain the body. But this repair is not isolated. It is part of a larger network. Hormones are circulating. Metabolism is being regulated. Glucose is being managed. Insulin is being balanced. The endocrine and metabolic systems are working together. They coordinate the rebuilding. They provide the resources. They maintain the environment. That coordination is vast, complex. It extends beyond growth hormone and cortisol. But that comes next. For now, there is only this. The slow waves, the GH pulse, the protein synthesis, the connective tissue remodeling, the unfolded protein response, the quiet, heavy work of deep sleep. The muscle is being rebuilt, slowly, carefully, in the dark. Everything here is becoming stronger. Chapter 6. Sleep, Endocrine, Metabolic Regulation and Recovery. The night continues. Deep sleep has passed. Growth hormone has pulsed. Protein synthesis is elevated. The muscle is rebuilding, but the work is not finished. Hormones are circulating, quietly, steadily. They carry messages, they set rhythms, they coordinate systems that span the entire body. These are not dramatic signals, they are timing signals. They tell the body when to build, when to break down, when to store, when to release. Sleep shapes these signals. When sleep is present, the timing is correct. When sleep is absent, the timing is disrupted. We begin with cortisol. Cortisol is a hormone produced by the adrenal glands. It rises and falls in a daily rhythm. This rhythm is called a circadian pattern. The pattern is predictable, consistent. Cortisol is lowest at night. This is the nocturnal nadir, the point of deepest suppression. During the hours of deep sleep, cortisol remains low. The body is not mobilizing energy. It is not breaking down tissue. It is resting. In the early morning, cortisol begins to rise. It peaks shortly after waking. This prepares the body for activity. It mobilizes glucose. It increases alertness. It supports the demands of the day. This rhythm is important. The low point at night allows the body to shift toward anabolic processes, building rather than breaking down. The high point in the morning prepares the body for energy expenditure. Sleep loss disrupts this rhythm. When sleep is insufficient, the nocturnal cortisol nadir is blunted. Cortisol does not fall as deeply. It remains elevated at night. This creates a problem. Elevated cortisol at night promotes a more catabolic environment. Muscle protein breakdown increases. The balance between synthesis and breakdown shifts. The muscle is being dismantled when it should be rebuilt. Over time, this disrupted cortisol timing can undermine recovery. The body is working against itself. The hormonal signal is incorrect. The muscle cannot adapt as effectively. We move to insulin now. Insulin is a hormone produced by the pancreas. It is released when glucose enters the bloodstream, usually after a meal. Insulin signals cells to take up glucose, to store it as glycogen, to use it for energy. Insulin also promotes protein synthesis. It activates signaling pathways inside muscle cells. One of these pathways involves a protein called ACT. When ACT is phosphorylated, it triggers downstream processes that support growth and repair. Insulin sensitivity refers to how well cells respond to insulin. High sensitivity means cells respond efficiently. A small amount of insulin produces a large effect. Low sensitivity means more insulin is required to achieve the same result. Short-term sleep restriction impairs whole body insulin sensitivity. After just a few nights of insufficient sleep, the body becomes less responsive to insulin. Glucose uptake is reduced. Glycogen storage is less efficient. Sleep restriction also tends to blunt muscle insulin signaling. ACT phosphorylation decreases. The downstream processes are less active. The anabolic response is weakened. This has direct implications for recovery. After exercise, the muscle needs to take up glucose. It needs to replenish glycogen. It needs to activate protein synthesis pathways. When insulin signaling is blunted, the efficiency of post-exercise glucose uptake is reduced. Glycogen replenishment is slower. The muscle takes longer to restore its fuel reserves. The metabolic environment is less supportive. The recovery is compromised. There are other hormones involved, leptin and ghrelin. These hormones regulate appetite. They signal hunger and satiety. They influence energy balance. Leptin is produced by fat tissue. It signals satiety. It tells the brain that energy stores are adequate. When leptin is high, appetite is suppressed. Ghrelin is produced by the stomach. It signals hunger. It tells the brain that energy is needed. When ghrelin is high, appetite is stimulated. Sleep deprivation and sleep restriction shift these signals. Leptin decreases. Ghrelin increases. The result is increased hunger, a greater drive to consume food. This shift can change energy balance during recovery. The person may eat more. Energy intake may exceed energy expenditure. This can interfere with body composition goals. It can complicate recovery nutrition. The appetite signals are not just about food. They are part of a larger metabolic coordination. They reflect the body's assessment of energy availability. When sleep is disrupted, that assessment becomes distorted. We notice inflammation now. Sleep loss increases systemic inflammatory markers, interleukin 6, tumor necrosis factor alpha, C-reactive protein. These are cytokines and acute phase proteins. They are produced during inflammation. When sleep is adequate, these markers remain low. The immune system is active but controlled. The inflammation is localized, appropriate, temporary. When sleep is insufficient, these markers rise. The inflammation becomes systemic, chronic, uncontrolled. Chronic inflammation contributes to insulin resistance. It interferes with insulin signaling. It reduces glucose uptake. It promotes metabolic dysfunction. Over time, this can lead to metabolic syndrome, a cluster of conditions that increase disease risk. A worsened metabolic environment undermines training adaptation. The muscle cannot respond to training stimuli as effectively. The improvements in strength and endurance are blunted. The body is fighting inflammation instead of adapting to exercise. Recovery requires a controlled inflammatory environment. Acute inflammation after exercise is necessary. It triggers repair, but chronic systemic inflammation is harmful. It interferes with adaptation. Sleep is the switch. It determines whether inflammation remains localized and temporary, or becomes systemic and chronic. We descend into the muscle cell now, into the nucleus, where genes are turned on and off, where transcription occurs, where the cell's internal clock resides. The muscle has a peripheral circadian clock. This clock is not the master clock in the brain. It is local, specific to the muscle tissue, but it is synchronized with the master clock. It follows the same rhythm. This clock coordinates nocturnal proteostasis. Proteostasis means protein homeostasis, the balance between protein synthesis and protein degradation, the clearance of damaged proteins, the folding of new proteins. During the night, the muscle clock activates programs that clear misfolded proteins. It upregulates autophagy pathways. It supports quality control. It maintains the cellular environment. When the circadian rhythm is disrupted, these programs are perturbed. The nocturnal protein clearance is less effective. Damaged proteins accumulate. The quality control is compromised. Circadian disruption can promote muscle wasting over time. The muscle loses mass, it loses function. This is not just from lack of use, it is from disrupted timing. The internal clock is out of sync. The maintenance programs do not run correctly. Shift work, jet lag, irregular sleep schedules. These all disrupt the circadian rhythm. They desynchronize the peripheral clocks from the master clock. The muscle does not know when to clear proteins, when to synthesize, when to rest. The result is cumulative. Over weeks and months, the disruption accumulates. The muscle cannot maintain itself properly. The adaptation to training is impaired. We pause here, floating in the cell. The hormones are circulating. Cortisol should be low, but may be elevated. Insulin should be effective, but may be blunted. Leptin should be adequate, but may be decreased. Ghrelin should be controlled, but may be elevated. Inflammatory markers should be low, but may be rising. The circadian clock should be synchronized, but may be disrupted. All of these signals interact. They shape the metabolic environment. They determine whether the body can recover, whether it can adapt, whether it can become stronger. Sleep is the foundation. It sets the timing. It maintains the rhythm. It creates the environment where recovery can occur. Without sleep, the signals are wrong. The timing is off. The coordination is lost. The body struggles. With sleep, the signals align. The timing is correct. The systems work together. The body recovers. There is more to this. The fluid balance, the lymphatic drainage, the clearance of waste, the removal of debris, the physical cleanup that happens during rest. But that comes next. For now, there is only this: the quiet circulation of hormones, the timing signals that coordinate metabolism, the internal clock that maintains proteostasis, the foundation that sleep provides. The muscle is not just being rebuilt with proteins, it is being maintained by timing, by rhythm, by the coordination of systems that span the entire body. Everything here depends on the dark, on the stillness, on the uninterrupted hours of night. Chapter 7: Fluid Balance, Edema Resolution, and Cleanup. The night is deep now. The muscle is quiet. The hormones are circulating. The proteins are being synthesized. The metabolism is being coordinated. But there is something else happening, something simpler, more mechanical. Fluids are moving. We drift through the interstitial space, the space between cells. This space is filled with fluid, a clear liquid. It surrounds every fiber, every capillary, every structure. It is the medium through which everything is exchanged. Oxygen arrives from the blood. It crosses into the interstitial fluid. It diffuses to the cells. Carbon dioxide moves in the opposite direction, from the cells, into the fluid, back to the blood. Nutrients move this way. Waste products move this way. Signals move this way. The interstitial fluid is not static. It is a river, slow, invisible, essential. This river must flow. If it does not, waste accumulates. Pressure builds. Swelling occurs. The tissue becomes uncomfortable. Recovery slows. The movement of this fluid is governed by physical forces: pressure, concentration gradients, permeability. These forces have been studied for over a century. The original model was proposed by Starling. It described how fluid filters out of capillaries and is reabsorbed back into veins. That model has been revised. The revised Starling principle includes the glycocalyx. The glycocalyx is a thin layer of carbohydrates on the inside of capillary walls. It influences fluid movement. It changes the balance. In most capillary beds, fluid filters out continuously. It does not reabsorb back into the veins as much as originally thought. Instead, proteins and excess interstitial fluid return mainly via lymphatics. The lymphatic system is the drainage system. It collects fluid. It returns it to the bloodstream. It prevents accumulation. It maintains balance. Without the lymphatics, the interstitial space would flood. Pressure would rise. Edema would form. The tissue would swell. Function would be impaired. After exercise, this system is challenged. Exercise increases interstitial fluid and muscle. The capillaries dilate. Blood flow increases. More fluid filters out. Pressure rises into the interstitial space. At the same time, exercise damages tissue. Membranes become permeable. Proteins leak into the interstitial space. This increases the osmotic pressure. More fluid is drawn out of the capillaries. The swelling increases. This is normal. It is part of the inflammatory response. The swelling brings immune cells. It dilutes irritants. It creates space for repair, but it is temporary. The fluid must eventually drain. The lymphatic system is responsible for this drainage. Lymphatics are thin-walled vessels. They begin as blind-ended tubes in the tissue. These are called initial lymphatics. They have flap-like structures at their openings. When interstitial pressure rises, these flaps open. Fluid enters the lymphatic vessel. When pressure falls, the flaps close. The fluid is trapped inside. It cannot flow backward. Skeletal muscle contractions help this process. When the muscle contracts, it compresses the tissue. Pressure rises. The flaps open. Fluid is pushed into the lymphatics. When the muscle relaxes, pressure falls. The flaps close. The fluid moves forward. Stretch also opens the flaps. When the muscle is stretched, the interstitial space is deformed. Pressure changes. The flaps respond. Fluid enters the lymphatics. This is why movement aids recovery. Light activity, stretching, walking. These actions drive lymphatic draining. They clear the interstitial space. They reduce swelling. But movement is not the only driver. The lymphatics can also pump on their own. Once fluid enters the initial lymphatics, it moves into collecting lymphatics. These vessels are different. They have smooth muscle in their walls. They have valves that prevent backflow. The smooth muscle contracts rhythmically. It squeezes the vessel. The fluid moves forward. The valves ensure it can only move in one direction, toward the larger lymphatic ducts, toward the subclavian veins, back into the bloodstream. This intrinsic pumping continues even during rest, even during sleep. The collecting lymphatics do not require movement to function. They sustain flow automatically. This is crucial. During the night, the body is still. There are no muscle contractions, no stretching, but the lymphatics continue to drain. The interstitial fluid continues to clear. The cleanup continues. Sustained lymph flow is essential for clearing interstitial solutes from muscle overnight. Lactate, hydrogen ions, potassium, proteins, cellular debris. All of these must be removed. The lymphatics carry them away. The process is slow, gradual, but it is continuous. By morning, if sleep has been adequate, the swelling has reduced, the pressure has normalized, the tissue has returned closer to baseline. The immune system is involved in this process. During sleep, immune cell redistribution occurs. The number of circulating natural killer cells decreases. Monocytes decrease. Lymphocytes decrease. These cells are not disappearing. They are migrating. They are moving into tissues, into lymph nodes. They are positioning themselves where they are needed, where inflammation is being resolved. Sleep also enhances interleukin-2 production. IL-2 is a cytokine. It supports adaptive immune function. It helps coordinate the immune response. It supports the transition from inflammation to resolution. This pattern indicates active immune surveillance. The immune system is not resting. It is working. It is resolving the exercise-induced inflammation. It is clearing debris. It is supporting repair. Sleep creates the conditions for this work. The body is still. The metabolic environment is favorable. The hormonal signals are correct. The immune cells can do their job without interference. Sleep deprivation disrupts this. When sleep is insufficient, pro-inflammatory cytokines remain elevated. Interleukin-6, tumor necrosis factor alpha. These cytokines promote inflammation. They do not promote resolution. This shift toward a more pro-inflammatory profile may impair resolution. The inflammation persists. The edema lingers. The soreness is prolonged. The tissue does not clear as efficiently. The lymphatics are still working, but the signals are wrong. The environment is not supportive. The cleanup is slower, less complete. There is one more system worth mentioning, the glymphatic system. This is a clearance system in the brain. It is enhanced during sleep. It clears metabolic waste from the central nervous system. The glymphatic system operates through perivascular spaces around blood vessels into the brain. Cerebrospinal fluid flows through these spaces. It carries waste toward the lymphatic vessels in the meninges. The waste is drained away. This system is brain-specific. It has been well documented in animals. There is indirect support for its existence in humans. But it is not a muscle system. The effects of glymphatic clearance on peripheral muscle recovery remain speculative. There is no direct evidence that the brain's waste clearance system affects muscle tissue. The two systems are separate. It is mentioned here only for completeness. The glymphatic system is part of the nighttime cleanup, but its role is into the brain, not in the muscle. We return to the interstitial space now. The fluids are draining. The lymphatics are pumping. The immune cells are working. The swelling is reducing. The pressure is normalizing. The tissue is becoming lighter, clearer, less congested. The rivers are flowing. The waste is being carried away. The balance is being restored. This happens every night, automatically, quietly, without conscious effort. The body knows how to drain itself. It knows how to clear waste. It knows how to return to equilibrium, but it requires time. It requires stillness. It requires the uninterrupted hours of sleep. By morning, the work will be largely complete. The fluids will have cleared. The swelling will have reduced. The tissue will be ready for movement, ready for the day, ready for another round of work. But that readiness is not yet here. The morning has not come. The night is still present. The body is still draining. still clearing, still preparing. The transition from night to morning is gradual. The body does not wake suddenly. It shifts, it prepares, it signals readiness in stages. That transition is coming, but that comes next. For now, there is only this: the slow movement of fluids, the rhythmic pumping of lymphatics, the migration of immune cells, the resolution of inflammation, the clearing of waste. The muscle is being cleaned, drained, prepared, in the darkness, in the stillness, without interruption. Everything here is returning to balance. Chapter 8, Morning. Readiness after sleep. The darkness is thinning. The night has passed. Hours of stillness, hours of repair. The muscle has been rebuilt. The fluids have drained. The hormones have circulated. The systems have coordinated. Now the body begins to shift. Dawn is approaching. Not yet arrived, but approaching. The transition from sleep to waking is gradual, gentle. The body does not jolt into awareness. It drifts toward it. Light begins to filter. Through closed eyelids, the circadian system detects it. Signals are sent. The brain begins to prepare. Wakefulness is coming. Cortisol is rising. This is not the elevated cortisol of stress or sleep deprivation. This is the cortisol awakening response, the CAR. It is a discrete morning pulse, a time signal that occurs shortly after waking. The adrenal glands release cortisol. The levels climb sharply. They peak within 30 to 45 minutes after waking. This pulse is predictable. It happens every morning. It is part of the circadian rhythm. The cortisol awakening response serves a purpose. It increases alertness. It mobilizes energy substrates. Glucose becomes available. The body is prepared for activity, for movement, for the demands of the day. After training, an appropriate CAR is linked to perceived recovery and readiness. When the CAR is robust and well-timed, the person feels ready, recovered, capable. When the CAR is blunted or delayed, readiness is reduced. The body feels sluggish, unprepared. The CAR is not something that can be forced. It is automatic, but it reflects the quality of recovery. It reflects whether the body has had adequate rest, whether the systems have been restored. This morning pulse is a signal, a message from the adrenal glands. The night is over. The work is done. The body is ready to move. We notice the blood now. The plasma, the liquid portion of blood. It is changed overnight. Within approximately 24 hours after exercise, plasma volume can expand by 10 to 15%. This expansion is substantial. It is driven by increased plasma proteins, especially albumin. Albumin is produced by the liver. It holds water in the bloodstream. Fluid retention also contributes. The kidneys adjust their filtration. They retain more water. The plasma volume increases. This expansion has benefits. It improves stroke volume. Stroke volume is the amount of blood pumped with each heartbeat. When plasma volume is higher, the heart can pump more blood with each contraction. This improves cardiovascular stability the following morning. The blood pressure is steadier. The heart does not have to work as hard. The circulation is more efficient. Plasma volume expansion also aids thermoregulation the following day. More blood volume means better heat distribution. The body can move heat from the core to the skin more effectively. Cooling is easier. Temperature regulation is more stable. The body has prepared itself. It has increased its fluid reserves. It has enhanced its capacity to handle the next bout of activity. We move to the spine now, to the intervertebral discs. During the day, these discs are compressed. They lose water. They become thinner. The spine shortens slightly. This is normal. It happens to everyone. During the night, the compression is relieved. The body is horizontal. Gravity is no longer pushing the vertebrae together. The discs can rehydrate. They absorb water. They swell. They regain their height. Connective tissues throughout the body undergo similar rehydration. Tendons, ligaments, fascia. They absorb water during recumbency. They become more hydrated by morning. Hyaluronan accumulates into these tissues overnight. Hyaluronan is a large carbohydrate molecule. It holds water. It creates viscosity. It provides cushioning. But in the morning, this accumulation contributes to transient stiffness. The tissues are more hydrated, more viscous. Movement feels restricted. Joints feel tight. This is morning stiffness. It is not damage. It is hydration. The tissues are full of water. They need movement to disperse it. Movement changes this. As the body begins to move, the tissues warm. Heat increases compliance. The stiffness reduces. Hyaluronan is dispersed. The water is redistributed. The tissues become more mobile. This is why gentle movement in the morning is beneficial. It does not need to be intense. Walking, stretching, light activity. These actions warm the tissues. They restore mobility. They convert rustiness into smoothness. We focus on the joints now, specifically the synovial joints. These are joints that contain synovial fluid. The knees, the shoulders, the hips. The fluid lubricates the joint surfaces. It reduces friction. It nourishes the cartilage. During the night, the joint is still. The synovial fluid settles. It becomes stratified. The nutrients within it are not evenly distributed. The cartilage is not being loaded. It is not being nourished actively. In the morning, movement begins. The joint moves. The synovial fluid is mixed. It circulates. The nutrients are redistributed. The cartilage surfaces are exposed to fresh fluid. Loading also matters. When the joint is loaded, the cartilage is compressed. Waste products are squeezed out. When the load is released, the cartilage expands. Fresh nutrients are drawn in. This is how cartilage is nourished. It has no blood supply. It relies on this loading and unloading cycle. Morning movement and loading accomplish this. The synovial fluid is mixed. The cartilage is nourished. The initial rustiness is converted into smoother motion. The joint becomes more responsive, more comfortable. This is not repair. This is maintenance. It is the normal process of joint function. But it is important. Without movement, the joints remain stiff. The fluid remains stratified. The cartilage is not nourished properly. We shift to the nervous system now. The motor cortex, the spinal cord, the pathways that control movement. These have been quiet during sleep, but they are not unchanged. Corticospinal excitability can show favorable morning profiles. Corticospinal excitability refers to how easily the motor cortex can drive the spinal neurons, how responsive the movement pathways are. In many people, especially morning types, this excitability is higher in the morning. The nervous system is more responsive. Motor commands are transmitted more efficiently. Movement feels easier, more coordinated. Chronotype matters. Morning people show this pattern more clearly. Evening people may show the opposite. Their nervous system is more responsive later in the day. But for those who are naturally morning-oriented, the body is primed for movement shortly after waking. Corticomotor plasticity is also influenced by sleep. Plasticity refers to the ability of the nervous system to adapt, to learn, to refine motor patterns. Motor skills practiced before sleep can be expressed with improved coordination upon waking. This is motor consolidation. The nervous system has processed the movement patterns during sleep. It has refined them, strengthened the relevant connections. In the morning, those skills are more stable, more automatic. The movement feels smoother, more natural. This is not just practice. This is the benefit of sleep. The nervous system has done its work overnight. The body is ready to move, not just mechanically, but neurologically. The pathways are prepared, the connections are optimized, the coordination is available. We consider the inflammation now. The markers that reflect tissue damage and repair: interleukin-6, creatine kinase, C-reactive protein. These rise after exercise. They peak at different times. They resolve at different rates. Morning readiness depends on the training load. With moderate training, some markers may be declining by morning. IL-6 can begin to fall. The acute inflammation is resolving. CK and CRP may not yet be maximal on the first morning. They peak later, 12 to 72 hours after exercise. The body is in transition. The damage is present. But the acute phase is passing. The repair is underway. The soreness may be mild or absent on the first morning. It will likely increase later, but for now, the body feels relatively capable. With extreme training loads, the pattern is different. Soreness may already be rising, inflammation may be climbing, the markers may be elevated. The body is still in the acute phase. Readiness is reduced. More rest is needed. The difference is dose dependent. Moderate stress creates a window of readiness the next morning. Extreme stress does not. The body requires more time, more recovery, more cycles of sleep before it is ready again. This is not failure, it is physiology. The body communicates its state, it signals readiness or the need for rest. The morning after training is a moment of assessment. The body reveals what it has accomplished overnight. We pause here. Dawn has arrived. The eyes are still closed, but consciousness is returning. The cortisol pulse has occurred. The plasma volume has expanded. The tissues have rehydrated. The joints have prepared to move. The nervous system is responsive. The inflammation is in flux. The body is ready, or it is not. Either is acceptable, either is information. The muscle has been rebuilt. The energy has been restored. The fluids have been cleared. The signals have been coordinated. The night has done its work. What remains is movement. Gentle, gradual, the transition from rest to activity, from stillness to engagement, from sleep to waking life. But that is no longer the story of recovery. That is the story of living, of using what has been restored, of moving through the day with the capacity that the night has provided. The recovery is complete. For now, until the next training session, until the next bout of stress, until the cycle begins again. The body will be ready when that time comes. It knows how to recover. It knows how to rebuild. It knows how to prepare. It has been doing this every night, for years, for decades, for a lifetime. The muscle remembers, the systems remember, the body remembers. And when the work is done, when the training is finished, when the stress has been applied, the body will return to this, to the stillness, to the darkness, to the quiet processes that restore capability. This is recovery, simple, automatic, essential. Everything here has returned to readiness.